Hello, my name is Diva Whalen, and I'm from Meharry Medical College in Nashville, Tennessee. Has anyone ever tried to find where's Waldo? You know, Waldo, the one with the red and white striped shirt, glasses, beanie, traveler stick, red and white cap, that where's Waldo? Now, have you ever tried to find him during the Christmas edition? Oh my goodness, everyone and everything is either red or white or green or a combination, and it is exhausting. However, when you find Where's Waldo, you kind of want the satisfaction of plucking him from the page, putting him on a trophy stand, outlining the edition that you found him in, the date, and probably how many hours it took you to find him. That's how much satisfaction we want from Where's Waldo. But most of the time we circle him and we try to continue on to the next Where's Waldo search and just continue. Well, I have just described to you triple negative breast cancer, or TNBC. TNBC is a breast cancer with outlying features, distinct characteristics, and defined biomarkers. And just like in the search of Where's Waldo, doctors and research scientists like myself, we can describe TNBC to a T. We can outline and pinpoint clusters of TNBC cells and even identify a tumor mass. But just like in Where's Waldo, we cannot remove TNBC without causing harm to the rest of the body. And it's because TNBC has no direct target therapies. So we treat cancer patients with TNBC with chemotherapy drugs. And chemotherapy is just drugs that generally treat but not specifically target a cancer. They do not specify or go straight to a problem. So therefore they affect healthy cells as well. And that is why I study triple negative breast cancer. Because triple negative breast cancer primarily affects African American, African and Latino women. The women have to suffer through chemotherapy with no hopes of direct targets. And my research is focused on not only TNBC, but a biomolecule called RAS-GRF2. Now, RAS-GRF2 is abnormally expressed because it's only supposed to be seen in the brain and in fetal development. So what is RAS-GRF2 doing in this triple negative breast cancer? Well, it is acting like the finger that cuts on the light switch. And that light switch, what's turned on, is very hard to turn off. And many chemotherapy drugs do not focus on cutting it off. Therefore, the women progress quickly and the diagnosis is fairly poor. Yet, my research in TNBC on RAS-GRF2 provides hope and highlights this biomolecule as probably a possible answer and target for triple negative breast cancer. Because we, as research scientists and doctors, can use TNBC or can use RAS-GRF2 as a potential biomarker and feature that finally identifies Where's Waldo. But unlike the other characteristics of TNBC, RAS-GRF2 is very particular to triple negative breast cancer. It's just like if Where's Waldo decided to wear a rainbow hat instead of his traditional white and red beanie. This would stick out because cancer cells have RAS-GRF2 present and active, whereas healthy cells don't have RAS-GRF2 present and active at all. So while we continue in our quest to find the Where's Waldo of triple negative breast cancer, I would charge you to participate in breast cancer studies, constantly and regularly eat your fruits and vegetables, exercise for at least 30 minutes a day, and remind a woman that you know, or even yourself, to have a self-monthly breast exam. And this will help in our fight against TNBC. Thank you.